Welcome to the Diabetes Bio Podcast, a podcast for the Diabetes Journal, the flagship research publication of the American Diabetes Association. Each month, we'll interview one or more of the authors from the feature articles, the papers of the month, or other articles published in that month's issue of the Diabetes Journal. We also have a Sweet Talk segment of the podcast, which is more of an open format discussion of additional articles, conferences, or major events in the field that we'd like to highlight. My name is Darlene Sandoval, and I'm joined by my podcast co-host, Dr. Kirk Habiger and Dr. Kevin Williams. How are you guys doing today?
Hey, Dar. Kevin?
Yeah, doing well.
What do you guys think? You think we can get Tade Pagacha to do an interview with us for the Diabetes Bio podcast? We could talk about his training principles.
That would be awesome. That would be very cool.
Heck, I'd settle for Jonas, man. April 16th in the hospital, and now he's second in the Tour de France. That's amazing.
So it was an amazing recovery. Yep.
All right. Well, let's talk about, I guess we have to get some work done. So let's talk about today's episode. Kirk, what do we have in store for our listeners this week?
Thanks, Dar. This month, we'll talk to Dr. Danwei Huangfu, who is a professor and member of the Sloan Kettering Institute of Memorial Sloan Kettering Cancer Center and Weill Cornell Graduate School of Medical Science. She's joined by Dr. Inbal Caspi, who was a graduate student in her group, and she's now an adjunct assistant professor at Pace University. They'll be chatting with us about their manuscript titled Glucose Transporters Are Key Components of the Human Glucostat. We'll also hear from Dr. Deepthi Mallela, who is one of the co-lead authors of an article titled Comprehensive Clinical and Genetic Analyses of Circulating Bile Acids and Their Associations with Diabetes and its Indices.
And Kevin, we have another, I think, special Sweet Talk segment. Why don't you tell us about that?
Yeah, really excited about this one. Today, we're talking with Dr. Bernard Torrens and Dr. David Hodson. They contributed an article to the Classics in Diabetes series that is based on work previously published by Dr. Torrens in the journal in 1993. In that work, Dr. Torrens cloned, sequenced, and functionally expressed the human GLP-1 receptor. This really was seminal work that has spearheaded many advances in GLP-1 biology, even going on to today and into the future. So be sure to tune in to later in the program.
All right, thanks. Let's jump right in. Next, we're going to discuss the paper of the month from the August 2024 issue of Diabetes, which is titled Glucose Transporters Are Key Components of the Human Glucostat. Today, we are joined by lead author, Dr. Inbal Caspi, and corresponding author, Dr. Danwei Huangfu. Welcome, Inbal and Danwei. Congratulations on your paper, and thank you for being a part of this month's podcast.
Thank you for inviting us. We are very excited.
Yeah, that was great. That was in tandem. Hey, we always like to start with an icebreaker. So we'll start with Inbal. Can you tell me your favorite fun fact about science? It doesn't have anything to do with this project, just something that you keep in the back of your head.
Well, I love being able to appear the smartest in the room whenever someone asks a biological questions. And I like, oh, well, did you know that in physiology, this and this, and immunization and this and that? So I like the background that it allows me to feel good about myself.
No, that's great. That's great. And Danwei?
I just came back from a meeting that's talking about human genetic variants. And when I talk about variants to my daughter, she's like, oh, Loki? It's a TV show she's watching. So yeah, variants can mean different things to people.
That's right. That's right. Thank you very much.
Nice. Danwei, we're going to start with you. And the first thing we like to do is just give our authors the opportunity to highlight any other contributing authors or collaborators to this work.
There are a few people in the lab who also assisted with the work. They are Julian Felicio, Pen Yang, Dingyu Liu, and Jilin Yan. So the paper is, of course, the outcome of teamwork. One of our major collaborators is John Odorico and his student, Daniel Tremel. John is a professor at University of Wisconsin. His lab works on also stem cell differentiation, but with a major focus on pushing for transplantation. So eventually we want to achieve, stem cell derived beta cells for transplantation as a treatment for diabetes patients. I would say John's contribution, a lot of it is actually perspective. My lab was basic biology, and John has firsthand experience with the clinical research. So he's providing a lot of the clinical perspectives. And in addition, they also helped us verify the GLUT1 and GLUT2 expression in fetal and adult pancreas tissue. So it's very satisfying for us to see consistent results coming from two different labs.
Yeah, that's really neat. And it's really neat to have that opportunity to have translation of your work, right?
Exactly.
Why don't you tell us a little bit about your research program, maybe a general overview, and then how it either led to this specific project or how this project fits in?
So my lab works on using human pluripotent stem cells for understanding the genetic basis of human disease and development. In particular, we love the pancreas and we are interested in diabetes. We are particularly interested in how to use the stem cell system to explain differences between human and mice, both in terms of their physiology, cellular function, genetic and molecular basis of disease. Actually, we have a long-standing interest in this area. Previously, we have investigated a gene called GATA6 in humans. When you lose just one allele of the two copies of the GATA6 gene, patients suffer from neonatal diabetes, so it's quite bad. In mice, they don't seem to care much when they lose just one copy of GATA6. So this is how we, started our journey trying to use the stem cell-based system to understand the difference between human and mice. And in fact, the word glucose transporters, these are the ideas are all coming from Inbal. Perhaps Inbal can elaborate.
When I started in Danwei's lab a long time ago, she does this thing that I love, which is giving scientific freedom to the student. So we get the opportunity to choose our project rather than be given it. And I was working on beta cells previously before I started grad school, and I was really interested in them. And we started talking about the differences between mice and humans and why do they have different glycemic set points. So once we started thinking about that idea, we started looking at potential differences. And one of those differences was the different array or profile of glucose transporters. So glucose transporters are a very big family of transporters, and different species express different types of glucose transporters in their beta cells. So we were wondering if this difference is in fact responsible for the change in glucose homeostasis and hence the glycemic set point between mice and humans. So it was a very interesting journey.
Yeah, that's a great introduction. So I guess to build off of that, Inbal, in your paper, you state that prior studies had relied on transplant into diabetic mouse models. I wondered if maybe you could tell us a little bit about your strategy and the specific mouse model that you chose and sort of caveats of that model and what it taught you about the biology of glucose homeostasis.
Yeah, so most previous studies that transplanted human islets into mice used diabetic mice as models. Either the beta cells were not properly functioning or they were completely ablated. And then they transplanted the islets in order to show that this can resolve the diabetic state and restore blood glucose level. So basically, the mice didn't have their own endogenous functional beta cells. And for us, it was very important to show that what happens when you have murine islets, murine beta cells that are properly functioning, and then transplant the human islets to work together. So this practice allowed us to show a sort of dominance in the sense that glucose homeostasis and obviously the effect on the glycemic set point. Because if the mice, they don't have their endogenous islets, but they only have the transplanted human islets, you can argue that the effect is because this is all they have. But if they have both types and we still see the reduction in blood glucose level, it's easier to argue that the presence of the human cells is the factor that contributed to this.
Yeah, and I think one of the strengths of your paper was your ability to separate, right, their endogenous insulin production from the transplanted islets. Maybe you can tell our listeners a little bit about that.
Yeah, so one of the important controls that we used in this experiment was to transplant murine islets as a control. Because again, one of the arguments can be it's the added mass of beta cells that would change the level of blood glucose in the transplanted mice. It's not the fact that it's human mice, it's just you have more of them. We had a previous inclination that when you transplant murine islets, that's not going to happen based on previous works that did murine islet transplantation. But it was a very important control. So when we transplanted an equivalent number of IEQs for the murine islets to the recipient mice, we did not see that big decrease in blood glucose level to the level that is comparable with human blood glucose. So we could conclude that it is the presence of human islets and not just more islets that facilitated this difference.
Very cool.
Yeah, I want to add that it's fascinating to see that when you transplant the human islets, right, you sort of effectively humanize the mice in some way so that their glucose set point actually drops to about the human level. So that suggests to us that the islets is probably the beta cells within the islets. They really are the major components to the glucose set point.
Yeah, that's really interesting. In your introduction, Danwei had mentioned the fetal and adult islet studies. So, Inbal, can we talk a little bit more about those studies? It seems like you have some differences in the relative expression of the glucose transporters, and we're wondering whether that has any implications for glucose regulation in the fetus versus adult.
Yeah, so as Danwei mentioned, this work was performed by Dr. Daniel Tremmel from the Odorico Lab when he was a PhD student there. And I think it adds a very important and interesting aspect to the work. We were very lucky to have the ability to actually look at human fetal pancreas. And from the analysis of the images and stains that he took, we could actually see that there is a difference between the expression profiles of GLUT1 and 2 in fetal compared to adult human islets. So in the fetal, what we did see that there is a much bigger population of GLUT2 expressing insulin positive cells, so beta cells, compared to the adult where the majority of the cells are GLUT1 positive. The co-expressing population, so GLUT1 and 2 expressing cell, is also much bigger in the fetal islets compared to the adult. While we can't make definitive conclusions just based on expression experiments, this is a very interesting insight to how potentially glucose homeostasis is taking place in fetal time versus adult time. So we know that fetal pancreas and even neonatal pancreas is not performing to the extent of the adult. So that means it doesn't exhibit the biphasic insulin secretion pattern. It doesn't respond proportionally to changes and fluctuation in extracellular glucose. So it doesn't properly regulate the blood glucose level. And that changes with time and maturity of babies when they do exhibit an independent mature response of insulin secretion to changes in glucose fluctuation. And that kind of coincides when we start seeing a mature profile expression pattern of the glucose transporters. So it's very interesting to see that there might be a connection between the expression profile of the transporters and the proper glucose homeostasis process that kind of gradually mature and gained from fetal time to an independent baby.
Yeah, maybe I want to just add that, this GLUT2 expression in fetal beta cells is something that most people sort of overlook. We typically think about GLUT1 as the predominant glucose transporter in human beta cells, whereas in mice, GLUT2 is the predominantly expressed glucose transporter. Now, previous studies, people actually observed that GLUT2 deficiency, so GLUT2 homozygously mutated people, they showed transient diabetes phenotypes. So we think actually, even though we don't have direct functional proof for what's actually happening in humans, the fact that GLUT2 is expressed during fetal development could potentially explain why GLUT2 mutated people, they actually show this transient diabetes phenotype. It's transient because in adults, there is GLUT1 that's taking over the job.
Yeah, that's really cool.
And not to get too far into the weeds, but there are differences in the capacity and the affinity for these transporters for glucose, correct?
Yes.
Can you comment on that and how it informs the set point, the glucostat?
So GLUT1 has a Km value of around 5 millimolar, and GLUT2 has a Km value of about 17 millimolar. So these Km values are when there is half occupancy of the transporter. This is when they work their best. And we know that there are ranges in normal glycemia in humans where there is a range that is considered rest or normal, and then there is a range that is considered fed. So you wouldn't have it normally unless you're like me and you like cookies, and then your blood glucose level is elevated. So one of the hypotheses that we have is that the difference in GLUT1 and GLUT2 and the difference in their Km values allows the cell to understand the changes in blood glucose levels. So routinely GLUT1 that has the Km value of roughly normal blood glucose level shows that when the blood glucose level gets elevated, GLUT1 is in saturation. And then GLUT2 kicks in and starts to transport all the excess glucose. So it can reflect to the beta cell that there is more glucose out there right now, and you need to secrete more insulin to handle the situation. So that was like, to me, one of the most interesting things that came out of this project, that because it's so smart. It's so
very cool cellular physiology.
I know.
Yeah, absolutely.
Simple mechanism to reflect this super important change that has the potential to affect the entire physiological processes in the body. Yeah, to me, it's very cool.
So another thing that I thought might be interesting was that there were some pretty profound differences in the random or ad libitum glucose, depending on the transplant, right?
Yes.
When you humanize the mouse, you were able to reduce the set point in those mice, right? But glucose tolerance didn't quite follow the same profile. Can you comment on those and what that might tell us about the physiology of the mouse?
Yeah, so it's the main difference between the blood glucose level testing and the GTT experiment is the fact that the GTT mice were fasting for 12 to 16 hours before the experiment, while the monitored mice were not. We caught them at all stages. So again, it's less cohesive. The results are less cohesive. The point of the IPGTT experiment was to show the ability of the mouse to respond to an elevation in glucose by secretion of insulin and reducing the blood glucose level back to normal. In that sense, the important thing was to see that we get an elevation in blood glucose level following the injection. and then to see a pattern of gradual reduction in the blood glucose level. The fact that it's kind of compares in terms of between the human islets, transplanted mice, and the controls, to us, it reflects the fact that the transplantation did not hinder the ability of the pancreas plus the transplanted islets to efficiently respond to changes in extracellular glucose. It means that transplantation by itself, because we said you add masses of islets, how is that going to affect it? I mean, there was the possibility that it's going to do it faster, or we didn't follow kinetics to like a very fine point, but the pattern itself shows that the transplantation itself still facilitates proper beta cell response.
Yeah, and even the insulin levels that you measured were 30 minutes after the injection, right? So
Yes.
there's some definite potential dynamic range that could have been happening, at least in insulin during that time.
Yeah. Sadly, this is a very hard experiment to do.
No,
It requires so much blood. So adding more time points to the
absolutely.
insulin measurements.
Yeah, you had a lot of groups of mice.
Yes.
So you had some other cool sets of studies where you did a CRISPR-induced GLUT2 knockout with your transplant. So do you want to tell us about those experiments and what you gained from them?
So we did a GLUT2 knockout using CRISPR in human embryonic stem cells, and we used those cells after differentiation as to SC islets to look at how different expression profile of GLUT1 and GLUT2 affects beta cell functionality in terms of glucose uptake and in terms of insulin secretion by performing a dynamic GSIS experiment. For GLUT1, we did not make a knockout. We used chemical inhibition that blocks the ability of the transporter to transport glucose into the cell. We used an inhibitor that it was proven to be very efficient and also very selective and specific for GLUT1. So we could play with absence and presence of either of the transporters. Again, we used the same aspect of looking at what happened at 5 millimolar and at 17 millimolar, like we talked about before, to see how the different transporters, if there is a difference in their effect in ways that correspond to their Km values and the suspected range of activities. That is nicely what we did see. So in 5 millimolar of glucose, we detected an increase in glucose uptake in the wild type SC islets, and for the GLUT2 knockout SC islet as well. Again, potentially because GLUT1 was still there to take in the glucose. But when we blocked the GLUT1, we did not see that elevation. Now, when we exposed the cells to 17 millimolars of extracellular glucose, we saw in the wild type an added effect. So more glucose was transported in. And in the knockout, we did not see that effect, which is a key result in showing that it's the presence of GLUT2 that had impact in addition of glucose uptake, and not just because you have more glucose. And again, when you added the GLUT1 inhibitor on top of the GLUT2 knockout, you can see a further reduction in glucose uptake. And we saw something very similar in pattern for the dynamic GSIS, so the insulin secretion measurement at 5 millimolar, the wild type secreted more insulin, and the GLUT2 knockout as well. However, at 17 millimolar, when you had the GLUT2 knockout, you did not get that additional insulin secretion. So these experiments demonstrate how each transporter is basically responsible for a range of extracellular glucose, and it mirrors the fluctuations and this change to the beta cells in order for it to properly secrete the amount of insulin required to lower the blood glucose back to normal.
Yeah, that was a really nice mechanistic piece to add on to the transplant physiology there. Danwei, I think I'd like to end with, giving you the opportunity to chime in again and tell us any key points or thoughts that you'd like to close with, or what's next.
Yeah, I have a few points I would like to highlight. One is the disease relevance. So GLU1 has been known for a long time to be the dominantly expressed glucose transporter in human beta cells. However, it's unclear whether human patients with GLU1 mutations would actually have diabetes, right? So that aspect is unclear. And GLU2, similarly, even though there are some patients identified, they only suffer from transient neonatal diabetes. So to us, extremely important that we conduct functional perturbation to understand the roles of these transporters in the human system. So what we are able to provide is really functional evidence for the requirement of both GLU1 and GLU2 in the human system. To me, it's also quite incredible that the mouse and human, right, they use different glucose transporters, that the Km values of these transporters match the blood glucose set point in these different species. So that's fascinating to me. Like Inbal was talking about, it's so clever. And in addition, the switch in humans from fetal to adult, right, in terms of the GLU1 and GLU2 expression levels, in some ways, it reminds me of the hemoglobin fetal versus adult switch, right? We're not sure whether, similar types of mechanism is in play to ensure the switch mechanism, but it's, nice to speculate about how the humans have adapted to have this kind of functional switch. So these are some of the open questions that we can evaluate in future work.
That's great. Thank you, Inbal and Danwei, for sharing your expertise with us today and talking about your recent work on the role of glucose transporters as a glucostat in humans and mice. I encourage the listeners to read the full article in the August issue of Diabetes. Thank you.
Thank you.
Thank you so much.
Next up, we're going to discuss the paper entitled Comprehensive Clinical and Genetic Analyses of Circulating Bile Acids and Their Associations with Diabetes and Its Indices. This is work from the August 2024 issue of Diabetes, and today we're joined by one of the co-lead authors, Dr. Deepthi Mallela. She is a staff research associate at the Cleveland Clinic. Welcome, Deepthi, and congratulations on your paper.
It's a great pleasure to be with you today. Thank you so much for this opportunity to discuss our work.
So, Deepthi, you say that you listen to our podcast, so you know that we always put the author on the hot seat by asking an icebreaker question.
Yes.
So, for you, I would love to hear something about how your personal life intersects with your work.
Certainly. So I want to start by saying that when I was 13, my father had a myocardial infarction. Subsequently, he had to undergo a coronary bypass graft surgery for a major 3-vessel coronary disease before he turned 40.
Wow.
So I was always curious as to why he suffered from heart disease despite having no risk factors. Like he didn't have any smoking, drinking, or hypertension, none of it. So this curiosity fueled my passion for preventive medicine, public health, and research, particularly from a young age. Additionally, we also have diabetes prevalent in most of my extended family, which is why I developed a very keen interest in researching the cardiometabolic diseases in particular. So I'm very delighted to be contributing to the scientific community through all my research work to make an impact on many such families like mine.
That's awesome.
Yeah, that's great insight. Thank you so much. We also like to start by giving our authors the opportunity to highlight any other contributing authors or collaborators that added to this work.
So first, I would like to thank my mentor, Dr. Stanley Hazen. He's a world-renowned physician scientist who is known for his pioneering discoveries in identifying major gut microbial-derived metabolites such as the trimethylamine N-oxide, TMAO, that predicts the future risk of heart disease, even in people who don't have any traditional risk factors. He's the corresponding author of this paper. I'm very grateful for all his guidance and support throughout this project. Second, I would like to thank our collaborator and the co-corresponding author on this paper, Dr. Human Alei at the University of Southern California Keck School of Medicine, who along with his team played a major role in performing the genome-wide association studies and also the Mendelian randomization analysis that we reported in this study. And finally, I would also like to especially thank Dr. Ibrahim Shuker, the co-first author of this paper, who is a clinical chemist and was a former member of the Hazen Lab, who laid the groundwork for this project by running the mass spec assays and all the human samples reported in this study, and also for contributing to the writing of this manuscript. Ibrahim, along with Dr. Ina Nemeth, our collaborator, who is an assistant staff at the Cleveland Clinic, developed the highly quantitative mass spectrometry method that we used for quantifying various bile acids in this study. This bile acid quantification method was described in much detail in an earlier method paper, which was published in the Journal of Lipid Research. I again want to thank Ibrahim, who is the first co-lead author of this paper, for performing all of the bile acid mass spectrometry assays and for helping in writing this paper. This work wouldn't have been possible without everyone's contributions, so I would like to thank them all.
Wonderful, wonderful. So you mentioned that you did this work as part of Stanley Hazen's lab, and maybe you can start by giving us sort of an overview of his research and then how it led to this specific project.
So I did this current work as part of the Hazen Lab. As you introduced, I'm a physician investigator leading the bile acid studies here in the Hazen Lab at Cleveland Clinic. My current research project is part of a multi-million-dollar program project grant titled Gut Microbiota and Cardiometabolic Illnesses, funded by the National Institutes of Health. So I want to give a brief overview of the work that we do at Hazen Lab. So at Hazen Lab, we mainly look for chemical signatures in the blood that predict the future risk of disease. And particularly, we focus on those that are of gut microbial in origin. We use an untargeted metabolomics approach to screen for both known and unknown metabolites that track with different disease phenotypes that we're interested in, such as cardiovascular disease, heart failure, diabetes, obesity, and so on. So we do this first in an initial discovery cohort. But once we identify the metabolite that consistently tracks with the disease, we then study this metabolite by using human clinical samples. So we determine whether these metabolites just merely coincide with the disease or actively contribute to the disease process. We do this by performing cell-based assays, animal models, etc. So this approach has led to some of the seminal discoveries of gut microbial-derived compounds, which we found in our Hazen lab that were linked to cardiovascular disease, such as the TMAO that I mentioned earlier, comes from the red meat consumption, and then phenylacetylglutamine, and the most recently discovered terminal niacin metabolites such as 2-PY and 4-PY that are published in Nature Medicine. So one such important partially gut microbial derived, I would say, that tracked with disease in the initial discovery cohort that we looked at with a small cohort of 80 subjects was bile acids. So bile acids has been my area of research focus ever since I joined the Hazen lab. For those audience who do not know what bile acids are, I would like to just say that bile acids are end products of cholesterol metabolism, and they're important signaling molecules that are synthesized in the liver. So they're first synthesized as primary bile acids, and then they are transformed in the intestine to secondary bile acids by, let's say, they get conjugated with amino acids such as taurine, glycine with the help of gut microbiota, and then they are stored in the gallbladder in the form of bile. So when we eat a meal, the gallbladder contracts, secretes the bile into the intestine, where the bile acid digests and absorbs these cholesterol and lipids in the food that we eat. So traditionally, we only know bile acids as important molecules that play a role in lipid and fat metabolism. But recently, now they're appreciated as metabolic signaling molecules, which regulate the glucose metabolism as well. I'm not going to go into the details, but I just wanted to highlight two particular roles of bile acids as signaling molecules. This might be of interest for the diabetic podcast audience. So the recent studies found that bile acids, they bind to the TGR5 receptor in the intestinal luminal cells, leading to the GLP-1 secretion. And then also similarly, they bind to the TGR5 receptor in the pancreatic beta cells, which triggers the intracellular cascading signals and promote the secretion of insulin release. So by playing these two roles, they contribute to the glucose homeostasis. So my sole research mainly focuses on understanding the role of these bile acids in various cardiometabolic diseases like diabetes, insulin resistance, obesity, and cardiovascular disease. So that's basically the work that we do at the Hazen Lab, and specifically my interest.
That was an excellent overview. I think that'll be really helpful for a lot of our readers. Let's get into some of the more nitty-gritty details of the current manuscript. So, in the introduction to the manuscript, you talk about the fact that there are challenges to measuring the different species of bile acids. So, can you talk a little bit about why this is challenging and how you got around that in this paper, or how you get around it in your lab?
Sure, that's a great question. So, when compared to the various other metabolites that we quantified here in the Hazen lab, there are some specific challenges that we noted while quantifying the bile acids. I want to highlight like 5 main challenges. So, the first one is bile acids have very diverse physicochemical properties, especially some of them being extremely hydrophobic and they have limited solubility in water. Second, the bile acids are found in very low concentrations in circulating plasma, and as such, they require a very highly sensitive method for quantification. Then the third one is the complexity of the plasma matrix itself. So plasma contains proteins, metabolites, lipids. So all these constituents of plasma can potentially interfere with measuring the bile acids. And the fourth is the structural similarity of bile acids. So there are several of the bile acids, and probably you must have seen, like we highlighted 60 of them in the paper. All of these bile acids have a steroid nucleus with 4 fused carbon rings. All of these have similar chemical properties, but many of them, they exist as stereoisomers, which means just like right and left-handed gloves. So they're very identical in structure, but have different physiological properties. And the last challenge is to simultaneously measure over 50 bile acids, especially if we think of the traditional conventional techniques that we already have. In this paper, we overcame those challenges by using some specialized sample preparation techniques and quality control measures. We used the stable isotope dilution internal standards for the bile acid molecular species to minimize the plasma matrix effects. And then we used the validation methods, such as assessments of both intraday and interday precision and accuracy, to see that there's very low coefficient of variation. And all the samples that we ran included the quality control samples to monitor the assay performance. Also, we used the liquid chromatography tandem mass spectrometry method, which allowed us to simultaneously quantify over 50 different bile acids with high specificity, including those stereoisomers that I was talking about. So overall, we were able to develop and validate a very robust and highly reliable method to quantify nearly 60 structural distinct bile acids across different biological matrices, such as mice plasma and human plasma. And also, I want to highlight that these bile acids span a concentration range from nanomolar to micromolar levels, very little in the circulating plasma. And I would say that out of the bile acids we quantified, like 30 to 40 bile acids consistently showed up, but there could be still additional bile acids that are still circulating, but not yet fully characterized.
Wonderful. So it seems you have access to a rather unique database to acquire these samples. Can you tell us a little bit about that repository and the subjects that you studied in this manuscript?
Sure, that's an important question, I think. So in this study, we analyzed a sub-cohort of the Cleveland Clinic Gene Bank Study cohort, which is a very large, single-center prospective cohort with nearly 10,000 participants, I would say. So the cohort consists of sequentially followed clinically stable subjects who were enrolled between the years 2001 and 2007 with no evidence of any coronary artery disease at the time of enrollment, but they underwent diagnostic cardiac evaluation at the Cleveland Clinic. So these subjects are prospectively followed for the development of any future cardiac events, such as coronary artery disease, percutaneous coronary intervention, or revascularization or death. So the two main goals of the gene bank were to learn more about the causes of the coronary artery disease and also to develop new and more improved treatments for the coronary artery disease. So we examined a sub-cohort of the gene bank cohort here in this study. So it consisted of 2,145 individuals who provided us the fasting blood samples at the time of cardiac catheterization. So we quantified almost 60 bile acids in the fasting plasma samples of these subjects, and then we analyzed to see how they are correlated with diabetes, insulin resistance, and obesity, which we described in this study.
Excellent.
Wonderful.
Let's get into more details about the paper. So you looked at the different types of bile acids in patients with diabetes, and one of the, I think, unique identifiers was that you were able to compare patients that were being treated for their diabetes and versus patients that weren't treated for their diabetes. So can you tell us a little bit about those results and what differences you saw and what the implications for those findings might be?
Yes, definitely. As I said earlier, the bile acids that we measured in this study, they were nanomolar concentrations in circulation, and they spanned across almost 4 orders of magnitude. So we noticed significant differences in the concentrations of specific bile acid groups among the subjects who had diabetes versus those who did not have diabetes. So to understand the results of our study better, I'll talk a little bit about the biosynthetic pathways of the bile acid metabolism. So in a nutshell, bile acids are synthesized by 2 major pathways called classical pathway and alternative pathway. So in the classical pathway, the cholesterol undergoes a series of hydroxylation reactions and synthesizes cholic acid, which is an important primary bile acid, via the 12-alpha hydroxylation reaction, which means the hydroxyl group is added at the 12th carbon of the bile acid, and hence it's called a 12-alpha hydroxylated bile acid. So similarly, the second primary bile acid, it's called chenodeoxycholic acid, is also synthesized in the liver by an alternative pathway. So the chenodeoxycholic acid undergoes the process called 6-alpha hydroxylation. After this reaction, it results in 6-hydroxylated bile acids, which includes hyocholic acid and its derivatives. So it was very fascinating to discover that in our study, the diabetic subjects had significantly higher levels of some of the 12-hydroxylated bile acids. In contrast, the diabetics also had very low levels of most of the 6-alpha hydroxylated bile acids that we measured. One particular bile acid that I want to bring your attention to is isolithocholic acid. It's gut microbially derived. It's a secondary bile acid, and it's found in very low concentration also in circulation among the diabetics, but it consistently showed up to be inversely correlated with both diabetes and obesity in all of our results. So because in the literature it is known that anti-diabetic medications can influence the bile acid composition, for this reason, we limited our analysis to just those subjects who were not using any antidiabetes medication at the time of enrollment and blood sample collection, for example. Even when we did those analyses, we found that after controlling for the medication use, we still discovered that the 12-hydroxylated bile acids were still positively associated with diabetes and insulin resistance, while the 6-alpha hydroxylated bile acids, along with the isolithocholic acid, were inversely associated with both diabetes and obesity.
Do you have any idea whether this hydroxylation process influences the way the bile acids might be signaling to either TGR5 or FXR? Does it affect the affinity?
So we do not have that information yet. Probably we will be able to identify that by conducting some mechanistic studies, which is one of our future goals. One of the studies that I read, I remember that the hyocholic acid does have a signaling pathway, which is the 6-alpha hydroxylated bile acid. It binds to the TGR5 receptor, and that's how in the pancreatic beta cells, it stimulates the insulin secretion. So there is a relation between having whether the bile acid is a 12 or 6-alpha hydroxylated and its effect on the signaling pathways, but we don't exactly know how it differs.
Yeah, if I can ask another real quick follow-up. In looking at those treated patients, did you see any changes in the 6-alpha or 12-alpha hydroxylated bile acids that correlated with any of the classic treatments? So metformin, GLP-1 receptor agonists, SGLT2 inhibitors.
Great question. I would like to mention here that we did not look at individual medication subtypes such as metformin, GLP-1 receptor agonists, or SGLT2 inhibitors in this study because we only had the information on yes, no to diabetes medication in the database we analyzed. But when we looked at the effect of anti-diabetes medication on the bile acid composition, the bile acid composition does undergo changes. So it's very interesting that it's not a generalized observation where we saw that all of the 6-alpha hydroxylated bile acids have increased or decreased. So because we looked at multiple structurally distinct bile acids, so some of them were increased, some of them were decreased. So it was very varied, but it did affect the bile acid composition, as we already know.
Oh, very cool. So you then examined if a recent GWAS study revealed any significant bile acid associations in loci for BMI and for diabetes. Can you tell us a little bit about what you learned there?
So first, I'll just define some terminology before I go into the details. So most of our genetic makeup is similar except for 1% that could be attributed to the single nucleotide polymorphisms. Single nucleotide polymorphisms are referred to as SNPs, which are very important and are defined as a difference in one single nucleotide between DNA sequences of two different individuals. So genome-wide association studies are an approach where we examine a full genome-wide set of SNPs in different individuals to see whether a particular SNP is associated with a disease process. So GWAS allows us to specifically study gene-associated loci that are involved in the disease. So all the subjects that we examined in this paper had genotype data available. So we performed genome-wide association studies and all of the significantly associated bile acids just to find out if there are any genetically determined host factors associated with the variations in the circulating levels. So out of the loci that were linked to the bile acids that were significantly associated with diabetes, we identified several loci linked to the bile acids that were significantly associated with diabetes. At these bile acid associated loci, the minor alleles at the lead SNPs were associated with increased bile acid levels in circulation. So we then used the publicly available large-scale GWAS meta-analysis data to evaluate and see whether this bile acid-associated loci were associated with BMI and diabetes. So the bile acid increasing alleles for three particular bile acids, deoxycholic acid, 6-ketolithocolic acid, and isolithocolic acid, they were correlated with diabetes and BMI. So we then performed two-sample Mendelian randomization analysis, where we studied whether the SNP that is associated with these increased bile acid levels was causally linked to diabetes and BMI. The results showed that the genetically increased deoxycholic acid levels were causally linked to the increased risk of BMI. Similar results we saw with the causal association between 6-ketolithocolic acid genetic levels and increased risk of diabetes. Most importantly, I want to highlight that the genetically increased isolithocolic acid levels were causally associated with the reduced risk of BMI and the reduced risk of diabetes.
That was great. Are there any other key points or thoughts you'd like to leave us with as we close the interview?
Thank you for the opportunity again to discuss our work on the podcast. I would like to emphasize to the audience that bile acids are very important signaling molecules that are associated with various cardiometabolic diseases, especially as we highlighted in this study, the diabetes and obesity, and also as I discussed in this talk. So bile acids can be useful treatment options in treating metabolic disease. For example, the ursodeoxycholic acid has been used for treating cholestatic diseases for several decades now. We are currently using the bile acid chelating agents in treating type 2 diabetes. And also, the effectiveness of bariatric surgery is also partially attributed to the impact on bile acid metabolism. So many great roles that bile acid plays in human disease processes. So one of our most consistent observation in the present studies have been the inverse relationship between isolithocolic acid and the various metabolic and cardiovascular phenotypes. So we hypothesized that some of these bile acids and isolithocolic acid in particular could be a novel treatment option for diabetes and obesity, whether if we supplement it directly or given as a prebiotic or probiotic after validation in animal studies. So to explore this further, we are planning to test the efficacy of bile acids in diabetes and diabetes-induced obesity mice models to establish and see if there are any biological relationships on how these bile acids contribute to the metabolic disease processes.
Interesting. We look forward to that.
Yeah, absolutely. Deepthi, thank you so much for discussing your work with us and for our listeners today. I encourage the listeners to read the full article in the August issue of Diabetes.
Welcome to another edition of Sweet Talk. I'm Kevin Williams.
And I'm Kirk Habegger.
Today we're speaking with Dr. Bernard Torrens and Dr. David Hodson. Dr. Torrens is an emeritus professor at the Center for Integrative Genomics of the University of Lausanne, and Dr. Hodson is a professor of diabetic medicine at the University of Oxford. They are both here to discuss the Classics in Diabetes article they contributed for this edition of the journal. It focuses on the work of Dr. Torrens to clone and functionally express the human islet GLP-1 receptor and also demonstrate that exendin 4 is an agonist and exendin 939 an antagonist for the GLP-1 receptor. This work was originally published in Diabetes in 1993. Thank you both for joining us today.
Thank you for inviting us.
Yeah, thanks for the invite, Kevin and Kirk.
It's good to have you guys.
We usually like to start with an icebreaker question. So to both of you, if you had not pursued a career in science, what profession do you think you might have been drawn to instead? Let's start with Dr. Torrens, and then we'll go with Dr. Hodson.
Well, I studied biochemistry in Geneva, but before starting that, I asked myself if this should be the best thing to do. And I was quite interested actually in literature, history, and philosophy. And I contemplated at some point going into this kind of study. But finally, going into something related to biology and study of living organisms was something that was more appealing to me.
Very cool.
For me, I actually trained as a veterinary surgeon originally, so the intention was to go and work with racehorses, in particular for assisted reproduction and loads of money and have a very nice life, but that didn't quite pan out. But actually, when I grew up as a kid, what I really wanted to do, I wanted to be an excavator driver and work in a gold mine or a construction site. Yeah. Because I was obsessed with heavy plant machinery, which I seem to have passed down to my son, actually, which is quite good because we need to.
It's in the genes.
Yeah, rescape the garden soon. So hopefully I can get him in an excavator.
Perfect.
Well, Dr. Hodson, can you start by maybe giving us a brief overview of the original study and then what motivated you to revisit the classic article on GLP-1 receptor?
Yeah, it's a good question. I mean, first of all, these classics series are a really good opportunity to dig around in the history of diabetes, which, we actually have one of the first editions in the library behind my office of the printed copy. So there's a lot of really important diabetes research that's gone into diabetes. So Bernard's work I really knew from kind of glucose transporter, which I think is probably most well known for, and his work in islet biology, and I kind of ignored more his brain-centric work until recently. But we started working on the GLP-1 receptor like 15 years ago in the islet, and then we started to kind of make mice models with the GLP-1 receptor. We began to do more heterologous cell systems and pharmacology, and all this time we were using this kind of construct for the GLP-1 receptor that I got from a company called CysBio, which actually was a clone of Bernard's original plasmid. So, GLP-1 receptor agonism is a really hot topic at the moment. It's the obesity drug. It's actually the first time I've been able to talk about my work down in the pub where, non-scientific colleagues will understand. And they'll generally ask me if I can, first of all, give them Ozempic, and then I will say no. And then they'll become interested about where Ozempic has come from. So, we're going to see a lot of this over the next kind of 10 years. But this hasn't really been the case for many of us working in the GLP-1 receptor field, right? There's about 60 years of really rigorous preclinical and clinical research that's gone into the development of these drugs. And sort of when I was thinking about the classics topic, I wanted something that was going to have really broad impact across the different domains of GLP-1 receptor agonist kind of development and clinical approval. So to me, the cloning of the human GLP-1 receptor, although it looks quite a simple thing to do now with the techniques we have available, led to this massive explosion in our understanding of GLP-1 receptor agonism. And I think Bernard will go into some of the kind of trials and tribulations of doing this at the time.
Yeah, that was great. Thanks.
Yeah, and building on that, Dr. Torrens, you touch on it a little bit in the article, but I wonder if you could take us back in time and give us maybe a brief rundown of the methods that you used in that original study to clone, sequence, and functionally express the GLP-1 receptor. What were maybe some key challenges that many today won't appreciate, and maybe some collaborations that were crucial along the way?
Yeah, sure. I think that there is the paper that was published in Diabetes, which was about the human GLP-1 receptor. But this was really for me, it was a follow-up on the work we had published a year before, which was about the cloning of the rat GLP-1 receptor. And maybe I should talk a little bit about that, because this was really, I think, the most challenging cloning and characterization of the receptor. At the time, this was in the end of beginning of the 1990s. So the cloning, the gene cloning, the molecular cloning techniques were very well developed around the world, except for membrane proteins. Membrane proteins represented a very specific challenge to clone them. And many techniques have been developed. And I was at the time in Harvey Lodish's laboratory at the Whitehead Institute. And in his lab, so they had started, before I came, to develop different techniques for the cloning of this membrane protein, and several different techniques have been used. Yeah, this is where I started, actually, by cloning the glucose transporter 2. But then there were new techniques that had been developed for what is called the expression cloning. So that means there is many ways when you clone a gene that you don't understand, you don't know. How do you know that it expresses a membrane protein? So the best way is to express the protein in a cell line and see, for instance, for the receptor, if it can bind the GLP-1 for the case we're interested in. So this was this expression cloning system that had been developed in Lodish's lab. And this was really the challenging part, because it required having all the RNA from a cell line or a cell type to be transformed into DNA, the classical cDNA cloning, and have this cDNA expressed in a cell, and then find a way to identify the cells which will express this specific receptor. And so there were techniques that we had developed to do that using GLP-1, and actually radioactively labeled GLP-1, which could bind to the receptor, and then you can identify by a microscopy. So it was kind of a tedious system to do that, but at the same time, extremely powerful. This had been developed with people in the lab when I was at the end of my postdoctoral stay here at the Whitehead. And so at the time, I knew after that, actually, because I was interested in GLP-1 because we knew it was an interesting molecule that could potentiate insulin secretion. This had just been characterized by Joe Ebner and Svetlana Moisov and Jens Holst in Denmark. And they had shown that this was a peptide that could stimulate insulin secretion very powerfully, but in a glucose-dependent manner. So which was something really, really interesting, and with the idea that it could be used as a drug. So this is why I embarked on this study. Well, one of the challenges, there were several companies like Novo Nordisk or Eli Lilly. They were already interested in cloning the receptor. And I knew they'd started, attempt at cloning the receptor, but I think they didn't start from the right material. And this is what made a big difference also. So they use cell lines, and cell line explain a variety of RNA for the receptor, a very big one, difficult to clone. And I started from rat islets. And this was also a very important aspect to isolate the islets and make all the process of cloning. So this was really the big challenge. In the end, I could clone the receptor, and then I characterized it fully when I was back here in Switzerland, when I started my own lab, doing the expression and testing the affinity for the GLP-1, the specificity of the binding, the signaling pathway, and so on. And this first part started for this was the rat. And this was really the most difficult or challenging part. And then came the cloning of the human receptor. This was obviously something important because while we knew from the work of others, from Jens Holz and Joe Ebner's lab, that GLP-1 can be used in human. It could be a glucoincretin also in humans, stimulating insulin secretion. But we knew also, this was described by Jens Holz and others, that it was very unstable, couldn't be used as a drug. There was a need to develop an agonist of the receptor which would be stable. To do that, it was important to have the human receptor because there are these small differences in structure, and you really need to have the human receptor to do that. So this is why, I embarked on the cloning of the human receptor, and the first tools that we had was the cDNA library that was from a human islet cDNA library that was produced by Mike Mockler at Washington University. But this one, we couldn't get a full-length cDNA from that. So we were in contact with people in Geneva who were developing human islet isolation procedure for transplantation. This was the group of Philippe Morel. And so we got islets from them and prepared our own cDNA library, and we could get the full-length cDNA library. So the cDNA for the human receptor. And then we could do the functional analysis. And of course, one thing that was interesting is we could test the exendin 4 and exendin 9. And so I had been in contact with John Heng, who was the person who discovered and first characterized exendin 4 and 3, the 939 form. He was in New York. And so he sent us these two peptides, and we could characterize them with the clone receptor. So we're absolutely sure that we are working on the receptor itself. And of course, this was something that was important collaboration. And then, as you know, the exendin 4 has been the first agonist for the treatment of diabetes. So these were, a few years of very exciting new data that we could generate through a lot of collaboration, actually.
That was a wonderful historical perspective there. I now have a question for both of you. This work began in the early 90s on GLP-1 receptor, and it obviously has far-reaching implications. In the article here, you highlight some of these implications, including pharmacology, signaling pathways, genetics, and structural biology. I'm wondering if maybe you can, build off of that first comment and tell us about some of the implications and the context of them now for our listeners. So let's start again with Dr. Dorrans, and then Dr. Hudson, you can chime in as well.
I think once you have a receptor, there are all the things that you have mentioned that could be studied that are important to understand the biology of the peptide, because that's the first thing that I've been interested in, is really the biology and the physiology of the receptor. So I was interested in what is the signaling pathway of the receptor that stimulates insulin secretion, also protects the beta cells against apoptosis, and so on. So we have done a lot of work along this line. But this is basics biology. But of course, there were all these other aspects that have been important for the pharmacology. And one, of course, was to devise agonist of the receptor. And the receptor is being used every day now by a number of biotech and pharma to develop a new receptor agonist. But maybe one thing to highlight is that to have the human receptor, we now know that this is critical, because one of the newly developed small molecule, which has been used in the clinic, the O4-glypron, works on the human receptor, but not on the rat receptor. So because there's one amino acid that makes a difference, and so it just highlights that. So, but I think maybe I should let David also talk, because he has been working on the receptor a lot, also in the signaling and other aspects.
Perfect. Thanks, Bernard. So it's quite funny because Bernard talks about the trials and tribulations of cloning the rat GLP-1 receptor, which was really difficult. And of course, nobody uses this anymore because, the human GLP-1 receptor is, the go-to receptor. We use them frequently in the lab, and we have all sorts of plasmids with different, kind of fluorophores labels on them, so we can pull down, do whatever you need with them. And of course, cloning has come a long way in that we still will do our own cloning. But if I was a pharmaceutical company, I would just pick up the phone and order a gene synthesis plasmid and get it done. So that's kind of the development. And alongside that, the GLP-1 receptor field has really developed technologically as well. So I think one of the really big things for me has been structural biology for the GLP-1 receptor, because it's not just about how the GLP-1 receptor works. It's actually provided a lot of insight into how class B G protein coupled receptors work. And it's become the kind of, representative model of a class B G protein coupled receptor. So other class B GPCRs are, the GIP receptor, the glucagon receptor, and so forth. And, using cryo-EM, you can now resolve what happens exactly to the receptor when the ligand binds, which residues interact to, cause these tiny changes in the motion of the receptor within the 7-transmembrane region, and then, of course, how it interacts with the G subunit. And although, that's quite high-powered technique, that's really discovered sort of the key residues which are needed for binding. So we're talking potency versus affinity, so how strongly something attaches to the receptor versus how efficacious it is. And it's really led to a lot of understanding about how the orthosteric site works, so that's the bit hanging out of the membrane, and the allosteric site, which is the bit sort of, kind of within the membrane, just sticking out, which acts as a modulator. And in fact, a lot of the breakdown products, will also have some effect on that. So it's been really eye-opening for me when I was doing the reading, because I'm not a structural biologist and I had to read a lot of these papers and actually learned quite a lot myself whilst doing that. And I think the really key thing there, and Bernard touched upon this, GLP-1 itself is really unstable. It's almost immediately degraded by, DPP4, which is on the cell surface, it's in the circulation. And, that really precludes using GLP-1 itself as a treatment for diabetes, because all these drugs were approved for diabetes, still are, before obesity. So it was by understanding sort of how we could truncate the peptide, N-terminal versus C-terminal, which was going to give the potency versus affinity and so forth, that we could actually come up, or Bernard could actually come up with colleagues for exendin 4. And all the other molecules have really, taken exendin 4 as a template. And Bernard showed this for the first time in his paper. And then they've modified that for either better plasma binding, or going forward now, with sort of different integration of agonists of other receptors, which I guess is a podcast for another day. So it has been huge. I think there's not a part of GLP-1 receptor biology that hasn't been touched by knowing the sequence. Such a simple thing to do, yet so important, because just imagine the scenario where we were still working in primary cells, which were really challenging studies to understand pharmacology. And these were brilliant studies, that predated Bernard's work. You just can't get the throughput and the drug screening aspects into that. I asked Bernard if he ever sort of made any money off this, but I think he'll answer that himself, actually, because I think that's probably quite interesting about the IP landscape.
If I had a good lawyer with me, maybe I would be in a different house now.
Nice. And as we wrap up, I have a two-part question for both of you. As we've talked about, the research area is a very hot topic now and going into the future. And I was curious, what do you think are the next steps in the field of research on GLP-1 receptors and GLP-1 biology? And then separately, any advice that you might give researchers, maybe advice specific to understanding G protein coupled receptors, or maybe more broadly advice for those that are just interested in pursuing a career in science?
There are two things, I think, to me. To have the sequence, the structure, the cryo-EM structure, as David has mentioned before, is really something fantastic that we couldn't, even imagine at the time we cloned that. And it helps for the GLP-1 receptor, but as David said, so there are the GIP receptor, the glucagon receptor, maybe the amylin receptor, and we know that there are pharma developing, small molecule or molecules that can activate all these receptors. So having the structure and learning about GLP-1 receptor, so we maybe be in a position to get better, more specific drugs or molecule that will target this different receptor and maybe in a different manner or different efficacy. And probably you don't need to have always very, very strong molecule like Ozempic to lose 15 or 20% of body weight. So you may do with different potency. So I think there is a lot to be done just considering this receptor. But the other thing is that we just don't know why why these drugs are so efficacious, why. So they need to have access for the control of body weight, for obesity. This is amazing, the efficacy. How does that work? So it's not fully clear. And still, there is something that is here. There is biology to be learned. Why is it so important, so active? Why can you not only treat obesity, but very positive impact on NASH and steatohepatitis and fibrosis and maybe also neurodegenerative disease. So it works, but we don't know how. So next generation should learn how it works, and this will be really fascinating to learn that. So.
Thank you both, Dr. Torrens and Dr. Hodson, for joining us today. Be sure to check out their Classics in Diabetes article in the August issue of the journal titled Building the Glucagon-like Peptide 1 Receptor Brick by Brick. This work revisits the seminal article by Dr. Torrens, Cloning and Functional Expression of the Human Islet GLP-1 Receptor, originally published in November of 1993. To our listeners, thank you for tuning in to this edition of Sweet Talk. Please follow, like, and subscribe for more Diabetes Bio content.
Thanks to all of you for joining us on the Diabetes Bio podcast. We hope that you're enjoying our discussion of the work that the Diabetes Journal is publishing each month. The feature articles we discuss in each episode are freely accessible at diabetesjournal.org/diabetes and are also listed in the episode notes for your reference. As with any podcast, it really helps us if you like, rate, follow, and subscribe to the podcast. And if you have any feedback for us, we'd love to hear that as well. We look forward to presenting to you each month.